This is note taker 7 on cell theory and structure. In this note taker, we'll look at the history of the microscope. We will talk about how cells were developed along with the cell theory that was created. And then we will take a look at the structures that make up both animal and plant cells, as well as their functions. When we reflect back to the What is Life note taker, we learned that all living things are going to be made of cells, whether it's one cell or many cells. Now, with a cell, it is the basic unit of structure and function of all living things. What that means is that the cell is the smallest unit that we have discovered that can be a living thing on its own. We know that there are things out there that are smaller, like atoms, but atoms are not living things. So the cell is the smallest unit that can be considered living, and when we put the cells together, they're the basic unit of structure of our living things. Now, with our cells, they are too small to be seen with the unaided eye. I like to use the term naked eye, which means no magnifying glass, no microscope. So around the 17th century, the microscope was invented, and it actually made the discovery of cells possible. The first microscope was actually created back in the late 1500s. It was a father and son that worked together, and they were spectacle makers. Spectacle is a fancy word for glasses. So they took two lenses and they put them on either side of a tube, and they realized that by doing this, it would magnify any object that you looked at through the tube. In the early 1600s, about 20 years later, Galileo built on the microscope design that they had created, and he added a focusing device, and basically that was just a way for the tube to be moved closer or farther away that allowed it to magnify better. It would focus so that it wouldn't be as blurry for people's eyes. You see here in the picture the first microscope, and you'll notice that there is no cord to an electric socket. There actually was a light. The light came from the candle, and the ball of water in the picture would concentrate the light and focus it down on the object that you were looking at. Anton van Leeuwenhoek also worked a little bit with microscopy, which is creating the microscopes, but he actually was the first to see and describe what he saw in pond water. And when he came up with these drawings, which you'll see on the next slide, people at first didn't believe him that these were living things. But as the invention of microscopes became more and more, as the microscopes became better and better, and more people started to see these, they realized, hey, actually, these things look exactly like what's floating around in a drop of pond water. Even though scientists were starting to see things using the microscope, it wasn't until Robert Hooke, who confirmed everything that van Leeuwenhoek had done with the microscope. It took Hooke to look at a piece of cork, which is dead plant material, and he looked at the shape, and you can see on the left-hand side picture, the left hand of the picture, it looks like they're little boxes. And he said that these look like little rooms called cells. So it wasn't until Robert Hooke that we actually got the word cells. Now, scientists continued to look at things under the microscope. More and more were using them. They were becoming more commonplace in labs to be used. And then through the 17th, 18th, and 19th century, there were a few scientists who kind of made some discoveries of more living things, not the dead plants that Hooke was looking at, but they looked at live plants, they looked at animals, and they came up with a couple of ideas for us. The first was a German botanist named Matthias Schleiden. A botanist is someone who studies plants. So he took pieces off of a live plant, where Hooke was using the dead plant material, but Schleiden took pieces of root, stem, leaf, flowers, and he looked at them under the microscope. And what he noticed is that no matter what part of the plant he looked at, they were all made of cells. About a year later, there was a German zoologist by the name of Theodor Schwann. He, kind of along with what Schleiden was doing, he was looking at different animal parts. He took skin samples, he took muscle tissue, he took blood samples. And no matter what part of the animal he looked at underneath the microscope, he saw that animals were made of cells. So these two gentlemen showed that, back in the time, people only knew really about plants and animals. They really hadn't done a whole lot of discovering any of the microscopic organisms yet. So far, we're looking at all the life forms on Earth, what they knew back then, being plants and animals, were all made of cells. About 20 years following that, there was a German physician named Rudolf Virchow. He came up with the idea that all living cells can only come from other living cells. So for something living to be, it had to come from something else that was also alive. Working together, along with some information and ideas from other scientists, they developed what was called the cell theory. You will need to know all three statements of the cell theory. The first statement is that all living things are made of cells. The second is the definition of cells. They are the basic unit of structure and function in living things. And the last one is that living cells only come from other living cells. The cell theory was used to disprove what was called spontaneous generation. Spontaneous means right now, generation means to make. So the idea behind spontaneous generation was that life could just poof appear out of nowhere. And back in olden days, people did believe in this because they would have a piece of meat sitting on a counter, because there was no refrigeration at that time. They would have the meat sitting on the counter, and a few days later, all of a sudden there are maggots crawling on the meat. The problem was, they saw the flies in the house, but they never saw the eggs the flies were laying. So one day the meat was clean, the next day there were maggots crawling on it, so they just thought, poof, the maggots must have magically appeared on the meat. But with their cell theory, we were able to disprove spontaneous generation and show that life does have to come from life. And there were two other famous scientists, Francesco Redi and Louis Pasteur, who did experiments to also disprove spontaneous generation. The first experiment was by Redi, and he wanted to disprove that maggots just poof appear out of nowhere. So he set up two sets of jars. The first jar, he put a piece of meat in it, and he left the top open. The second jar, which is on the bottom, he put a piece of meat in the jar, but he covered the jar with a piece of cloth and a rubber band so that nothing could actually get down into the jar. A few days later, he noticed that flies were attracted. The flies on the top jar could actually get in and land on the meat, but the flies on the bottom jar could only land on the top. A few days later, after the eggs hatched, he noticed that on the meat in the open jar, there were maggots, but on the jar covered with the cloth, the maggots were actually on the cloth. So by doing this experiment, he showed that maggots don't just poof appear out of nowhere, because if they could, then they would have been in the jar in the bottom picture. So he showed that life has to come from somewhere. You just couldn't see the eggs that the flies were laying. In Pasteur's experiment, he wanted to also disprove spontaneous generation, but he also wanted to show that by heating things, that can kill life. So what he did was he took this special flask that you see in the picture. It's called a swan neck flask because it has that neat little curve at the bottom. And he filled that with vegetable broth, like a vegetable soup type of liquid. And what he did was he heated that container. And once he heated it, you'll notice that in the little curve off to the right of the flask that there is some blue, because when you heat, you get evaporation of the liquid, and then when it hits that top of the flask, it cools down and slides to that curve. Well, once you have liquid there, nothing can get into the flask from the air. And he left it sitting for a few days. And what he noticed is that while that broth was blocking the pathway so no air could get in, what was in the main part of the flask, all that blue liquid, stayed nice and clean and clear. So then he wanted to show people that there was life all around you, you just don't always see it. So he took and broke the top of the flask off so that the air could get to the vegetable broth inside. And within days, the broth was all green and murky and nasty and full of life. So what he showed was two things. First, that when you heat stuff, it can kill stuff. It'll kill all the living stuff. And that life cannot just poof appear. But we do have life all around us that we can't see. Actually, his name, Pasteur, is what we now do to milk. We pasteurize it, which means when it comes from the cow, it's heated up to kill all those bad bacteria before it's then cooled down, put in containers, and sold to you in stores. For the remainder of the note taker, we're going to be looking at the difference between plant and animal cells. We're going to be looking at all the structures inside. And each of those structures are called organelles. It means little organ. Now on the left-hand side, I have a picture of an animal cell, and on the right-hand side, I have a picture of a plant cell. And as we go through, you're going to see that as we talk about each part, I will have a little portion of each of these pictures. If you only see the animal cell picture, it means that that part is only found in the animal cell. If you see only the plant cell picture, it means that that part is only found in a plant cell. So we're going to start on the very outside of the cell. And we're going to start with just the plant cell, because you can see just the plant cell picture at the bottom. Around the outside of the plant cell is a cell wall, and it provides support and protection for the plant cell. Now you'll notice that in red, I've highlighted part of this sentence. That is the function of the cell wall. I've purposely highlighted all of the structures. I've highlighted their functions in red. You will need that when you get to the cell project that I will assign. Now, the cell wall is going to be made of cellulose, and that's if you think of biting into celery, that fiber that kind of gets all stuck in your teeth, that is really strong and made of cellulose. So your cell wall is very strong, it's very rigid. It does have some openings to let things in, like water, oxygen, carbon dioxide, but it provides a safe barrier against the outside. And because it's so strong and stiff, plant cells can stack on top of each other almost like bricks. Now, going a little further into the cell, you'll see that I've now added the animal cell picture, because both the animal cell and the plant cell have a cell membrane. Cell membrane is a very flexible outer covering. So with the animal cell, it's going to need to be a little bit stronger than the plant cell, because the animal cell does not have a cell wall. So the cell membrane is its entire protection. But with the cell membrane, it is very flexible and can move, but it's like the bodyguard of the cell. It controls what is allowed to move into and out of the cell. So if you think of a dance club that you might have seen on TV, there's always that bouncer standing at the door, and your name has to be on the list before they let you in. This cell membrane is that bouncer at the door. If you're not on its list, you can't get in the door of the cell. On the inside of the cell, you have this ooey gooey jelly-like substance, and it's constantly moving. This is going to be your cytoplasm. The cytoplasm kind of holds all of the other organelles in place and allows everything to be able to move around the cell. Now, going further, more towards the center of the cell, we have a couple of cell parts here that are going to be found inside of the main part that I'm going to talk about. The main part is the nucleus, and you'll see a picture of the nucleus on the next slide. But the nucleus is going to be the control center or the brain of the cell. This is going to regulate all of the activities of a cell. Now, because the nucleus is so important, it gets its own membrane to protect it. So that's called the nuclear membrane. So it's going to be like a coating on the outside of the nucleus. And this is going to allow things to move into and out of the nucleus while it protects it. Inside of the nucleus, you have little structures called chromosomes. These are going to be your genetic material. When we study genetics second 9 weeks, we're going to be talking about why you look the way you do. And these chromosomes are what carry the traits that make you the way you are. These chromosomes are actually going to be made of nucleic acids. So if you've ever heard the term DNA, the Na portion means nucleic acid. So that's going to be your genetic material. In the very center of the nucleus, you have another little ball-like structure called the nucleolus. Scientists believe that that nucleolus is what makes ribosomes for our cell. And a little later we'll talk about what ribosomes are. So on the right-hand side below, you will see the plant cell. The purple is the nucleus. The dark purple in the center would be the nucleolus, and then you'll have the nuclear membrane surrounding the whole thing. In the picture on the left, you have the nucleus is kind of the, I don't know, it's like a grayish color. The nucleolus is the darker center, and the purple is the nuclear membrane. And in both pictures, you can kind of see some squiggles. Those squiggles would be the chromosomes. Leaving the nucleus, you're going to find the endoplasmic reticulum. Very long word, and yes, you do need to know what it means. But your endoplasmic reticulum in both pictures below, both in the animal cell on the right, animal cell on the left, plant cell on the right, the endoplasmic reticulum is going to be yellow. And these are a set of passageways. It's like a maze throughout your cell. And think of the hallways in the school, how you go from class to class through these hallways. Well, when something needs to move through the cell, it moves through these endoplasmic reticulum. On the surface of the endoplasmic reticulum are ribosomes. So in both pictures, they're the little black dots and blue dots that are on the yellow structure. These ribosomes are made up of RNA, which is another nucleic acid. That's what the Na stands for. And these ribosomes are going to be used to make protein. Protein is one of the building blocks of our body. It's what helps us grow and what helps us repair ourselves when we're injured. So protein, kind of important to have. Now, once the ribosomes make the proteins, they dump them into the endoplasmic reticulum, those passageways, and then those proteins get transported or moved around the cell to where they're needed. The little jelly bean looking structures on the animal cell on the left, they're blue, and then on the plant cell on the right, they are that kind of red and orangish color. These are going to be mitochondria, and they're going to be the powerhouses of your cell. This is where energy is released. So what is happening inside of this mitochondria is called cellular respiration. It's when they take the food molecules, they use a little bit of oxygen, they break those food molecules apart, and they release some energy for us. The more active a cell is, the more mitochondria it will have. So if you were to look at a cell in the skin of your earlobe versus a cell from your thigh muscle is going to have a lot more mitochondria because it's active in helping you walk around all day. The skin on your earlobe kind of just sits there, so it doesn't need as many mitochondria. Now the vacuole is a large round sac, and it can be used to store food, it can be used to store water, or it can be used to store waste products until the cell can get rid of them. if you notice in the pictures, they look very different between the animal cell on the left and the plant cell on the right. The animal cell vacuoles are very, very small, but you'll notice that the water vacuole that a plant cell has is very large. It actually takes up most of the plant cell. Can anybody guess why the vacuole in the plant cell might have to be that large? Well, think about what you do when you're thirsty. When you're thirsty, you get up and you walk to the water fountain. However, a plant cell cannot do that. A plant is held to the ground by its roots. So it can't just get up and walk across your yard to get some water when it's thirsty from the garden hose. So it needs a larger storage tank so that when it rains, it can absorb as much as it can and it can store it, because water is an important ingredient for photosynthesis. Moving on along, we get to the lysosomes, and you'll notice that the picture down below is just the animal cell. Lysosomes are very common in animal cells, but they're not as common in plant cells. Your lysosomes contain a chemical called an enzyme, and they have two jobs. The first job is to break down really large food molecules into smaller ones. Sometimes when nutrients are absorbed into our cells, the molecules are too big for the mitochondria to break down and release energy. So the lysosomes will break them into smaller pieces that the mitochondria can actually handle. The second job of the lysosome is the cleanup crew. As we grow, our cell parts need to reproduce and replace themselves. As the old cell parts die, or even when a cell itself dies, the lysosome releases all of its enzymes and it will digest the cell or the old cell parts so that it's cleaning up, so that we don't have all these dead cell parts or dead cells laying around. In the picture on this slide, you'll see kind of large green structures. These are the chloroplasts. They're green because they're filled with chlorophyll. Remember back to your photosynthesis note taker when we talked about the parts of a cell. Your chloroplast is the place in the leaf where photosynthesis actually takes place. The chlorophyll absorbs the sun's energy. Water and carbon dioxide also end up here in the chloroplast. That chemical reaction happens, and the plant will make sugar or food for itself. And chloroplasts, again, are only found in plant cells. The last part of the cell that you need to know is the Golgi apparatus, another big long name here. This is the picture on the left is the animal cell, picture on the right is the plant cell. They kind of look like stacks of colored pancakes. But your Golgi apparatus is used to package material to be transported through the endoplasmic reticulum. So if you think of the Golgi apparatus as the post office, it's packaging things up to be sent around the cell. Now, again, I'm going to remind you that all the red that I have highlighted in this note taker, you are going to need those definitions for your cell project that I will be assigning in class.